Now I get to introduce Yen, and I'll be very brief. So it is my absolute pleasure to introduce Yen-Lin Chen at the occasion of his B exam. And Yen joined my group in 2016, right? So it's 5 years in the group, which is, it's good for our average. 5 years is you know, kind of on the shorter side, which is great. Yen, I always like to just think back about how the students have been throughout their time here. And at the very start of his career in the group, he was open to exploring lots of different projects. And so Yen tried his hand at a lot of different things. And it took a little looking around, but once he found his stride, I think it's fair to say that he's been unstoppable. So, and you've carried me along for the ride, and I've learned a lot. So with that, I hand the floor to Yen. And are you able to share your own screen, or do I have to enable you?
I think so. Let me.
I just enabled you anyway, just in the event that I have to. So Yen, please.
So can you see the slides?
We can, and there's somebody in the waiting room, and it just vanished. Hold on. There we go. Got it. Okay.
Can you see the slide, the cover?
We can see the slides. Yes.
Okay, sounds good. So hello everybody. Thanks for coming to my exam. Today I'm very excited to tell you about my PhD work focusing on the wide-angle scattering of structured RNAs. So I will. Sorry, my computer kind of froze.
Yen, we're recording this. Do you know that?
I'm sorry?
We are recording.
Yes. Yes, I know. Yeah.
Okay.
Yeah. So I think they tell me to record if possible.
Okay, that's fine. I just want to make everybody aware. So.
Okay.
Okay.
So yeah. Okay. My computer works. So here's the outline of my talk. I will first give an introduction on the RNA and why we should care about RNA molecules. Then I will introduce a new method called wide-angle X-ray scattering to study RNA molecules and show you how the experimental data look like. And because this wide-angle X-ray scattering is a new and last employed experimental method, there will be unsolved problems, which will be the theme of this talk. I will talk about ways to model the experimental data with atomic structures of RNA and our discovery on the salt-dependent RNA duplexes. Then we will combine machine learning to decode experimental data and mine the useful information to understand RNA molecules. And I will talk about a new way to bridge molecular dynamics simulations with experimental measurement by using a strategy called sample and select. And finally, if we don't have any atomic structures in hand, I will propose a novel algorithm called frequency matching to get electron density from just the experimental data. So I will first talk about RNA. So ribonucleic acid, RNA is a negatively charged polyelectrolyte. RNA contains bases ACGU in the primary structure, which is a sequence. And these bases can pair in AU or CG via hydrogen bonding, forming secondary structures, including stem, loops, bulges, junction, hairpin, and so on. And the RNA secondary structure can further arrange themselves in three-dimensional space as tertiary structure, determining the biological functions or biological signals. And given the structural hierarchy, the RNA conformation can be extremely complex. So what does RNA do biologically, and why is it so important? The RNA performs coding and decoding in protein synthesis. And some RNA domains also have the functions of gene regulations and expression. And it also participates in biological reactions as ribozyme. And all of these biological functions require the RNA molecule to fold into a correct or active three-dimension conformation. For example, this TP5ABC RNA domain requires magnesium to fold properly into its native state. And lastly, RNA is very common in human, in living cell, and also the viruses. And some of the RNA domains are found to be disease-related. For example, this molecule called MALA1 are found to be cancer-related RNA molecule and has been the drug target in many studies over the past few years. So based on the number of base pair strands of RNA, we can simply categorize them into 3 classes. So the first is the flexible single-stranded RNAs. They are intrinsically highly flexible, adopting diverse conformations, and they exist in the solution as a conformation ensemble of different structures. And this single-strand RNA ensemble also can modulate the global conformation of RNA. And secondly, we have a rigid double-stranded RNA, which is formed by base pairing. And the base pairing reduces the flexibility and results in a rigid RNA duplex, which historically we call it A form. And this kind of A form RNA duplex structure was solved by X-ray crystallography, and due to its rigidity, it is a common assumption that it maintains this kind of A form conformation in the solution. And I will later tell you if this is correct or not. And finally, single-stranded RNAs might bind to RNA duplexes by forming proper base triples and further stabilizing conformation as RNA triplex. And the functionality of RNA triplex is still under studied, but the structure has been found across different species. So as you can see here, the structure of RNA has been so complicated just after 2 slides. And now let me tell you some of the structural technique we can employ to study those RNA molecules. Our method is known as solution X-ray scattering, which branches into small angle and wide angle. I will introduce small angle X-ray scattering first and move on to the wide angle X-ray scattering, which will be the main focus of this talk. So, for starter, I want to talk about small angle X-ray scattering, which is SAX, because this is a versatile and widely used experimental technique. And here is the schematic for experimental SAX experiment. And in SAX, we load the solution sample in the capillary and expose it with X-ray beam. Then we have an area detector to record the scattered X-ray photons. And because the molecule can rotate freely in the solution, the scattering is isotropic in the azimuthal direction. And by doing an azimuthal integration of the image pixels with respect to the beam center, we can obtain a one-dimensional solution X-ray scattering profile, which is I of Q versus Q. And I here stands for intensity. And the Q here is defined by the scattering angles and has unit of 1/Angstrom. And the higher Q you go, the higher resolution you will get. And we subtract the buffer scattering from the sample scattering and obtain this buffer subtracted profile for further analysis. And in SAX, we usually go up to Q equals 0.3, corresponding to about 20 Angstrom or 2 nanometers in the solution. As you can see, this is a low resolution technique. SAX tells you about the shape and envelope of the molecule. It reports a structural parameter called radius of gyration by taking the Q approaching 0. And also this is also known as the Guinea fit. And here's a cartoon showing a molecule and its RG representation. And the SAX also give you the probability distribution of pairwise distances by inverse Fourier transform, which might contain critical information of the RNA system. So for example, in this case, the black and green curves stand for rigid and flexible RNA systems, and they are very different. So in real application and measurement, SAX can be used to construct 3-dimensional shapes for comparison. So in the first example, we compare the reconstruction of RNAs to understand the sequence effect on the RNA junction. And when coupled with atomic structures generated by molecular dynamic simulations, SAX can be used can do more than reconstruction. And we can sample the structures that fits the experimental data and investigate the selected ensemble. And with this kind of idea, we found that this kind of small junction is modulated by the ion strength, by different concentration of the ions, and this short RNA junction with only 5 nucleotides can also shape the global geometry of RNA. In addition, SACs can be used to validate the solved structures by cryogenic electron microscopy or X-ray crystallography by docking the models and comparing the SACs profile. And this is another example where we use SACs to validate the atomic structures and behavior of an RNA triplex domain. So yeah, till this point, SAC is great in this, all these applications to understand RNA systems. However, what we actually get is a low resolution envelope or 3-dimension dummy atom reconstruction. And there might be ambiguity in docking or modeling. And this is the very inspiration that we need higher angle or higher resolution than SACs. So which means that we need to achieve higher Q or higher scatter angles. So higher or wider scattering angles can be achieved by moving the detector closer to the sample cell from 2 meters to ½ meter in WAXS, recording scattered X-ray photons at the wider angle. And the experimental setup is very similar to SACs. And this is the image taken by the Eiger X1M detector, and we perform the thing as a mutual integration and buffer subtraction. And we have the wide angle X-ray scattering. But you might wonder, we move the detector all the way closer to the sample cell only to get all these all these zero signals. But actually, in fact, if you plot it in the log Y scale, you see that the signals are not zero. These wide angle regimes contains the small signals or the fine features of the molecule in higher resolution. And in this case, we achieved Q equals 1.5, corresponding to about 4 angstrom resolution in real space, about 5 times sharper than SACs. So this WAXS profile is the RNA X-ray fingerprint containing unknown but rich structural information of the RNA system. So small and wide angle X-ray scattering experiments are usually conducted at the same time. And here's the setup at G1 chest in 2017, and we have the incident X-ray beam from the upstream and the sample cell sitting in the vacuum. And the wide angle detector is very close to the sample cell right here, and the small angle detector is further downstream. And similar setup is used for the Lick spin line at National Synchrotron Light Source 2 at Brookhaven National Lab. And the wide angle detector sits in the vacuum as the panel D, C, and D here, and they can record the water peak, which sits at Q equals 1.5 to 1.8. And with the robot arm, nowadays, the data acquisition is relatively straightforward. However, down to the molecular scale, there are many things to consider for computing this WAXS profile theoretically and precisely. So as I mentioned, the WAXS profile is very unique for different conformations with higher than 4 angstrom resolution. And therefore, the forward modeling requires consideration of the solvent and ion layers because they also contribute to the signal as well. And we apply the Debye formula and orientational average, considering all the atoms in the system, including explicit waters and hydrogens. And this computation scales linearly with the number of scattered angles and also number of RNA conformations. The complexity is kind of overwhelming, and I use Julia language on the Cornell Red Cloud for high throughput computation. And also with like small tricks with symmetry and some approximation, it takes about 10 seconds per conformation. So, so I mentioned that the WAXS profile contains structural information, which is mapped to one-dimensional profile in a nonlinear way via the Debye formula. And different from the SACs, there's no single parameter we can derive directly from the WAXS profile, and there's no method to interpret the data at all. So we need the help from atomic structures. So with atomic structures, we can forward compute the theoretical WAXS profile. And with theoretical and experimental WAXS profile, we want to know what kind of information we can learn. And also, we also want to know how we can bridge RNA conformations with experimental WAXS measurement. And finally, we also want to improve the MD simulations using the experimental WAXS profile. And those are the cases with atomic structures, but what if there is no atomic structure at hand? We want to know what kind of insight we can gain from just the experimental data and how to do that. So I will first talk about an extended ensemble optimization method and our discovery on the Saw-dependent RNA duplexes. In order to model the experimental WAXS profile, we collaborated with Albert Group at UT Austin and used molecular dynamics simulations to sample the RNA duplex conformations. And these sampled conformations form a conformational pool. For each conformation, we can forward compute the WAXS profile and see which conformation agrees with the experimental WAXS profile. And this process is done by a genetic algorithm selecting conformations from the pool by minimizing this new log chi-square statistics designed specifically for WAXS. And I call this extended ensemble optimization method because we extend the original EOM in the small angle domain into the wide angle domain. And we can assess the robustness of the selection by 5-fold cross-validation. So here I would like to show the selection result and the implication. Remember in the introduction, I mentioned that the RNA duplex is thought to be a rigid A form in the solution shown in the green curve and green duplex. However, as we measure the conformation in potassium, the WAXS profile suggests otherwise. And the blue duplex is a selected structure. Moreover, in magnesium, the duplex has another different conformation shown in red. So now we see that the RNA duplex is shaped differently by divalent and monovalent ions. And contrary to the general assumption, RNA duplex is not an A-form duplex in the solution. And from these structures, the duplex, we further analyze the duplex and see that the duplex is compressed and shaped by the magnesium ions, reducing the major group width as shown in the magenta cartoon model. And also the duplex unwinds a little bit. So this kind of magnesium-induced twisting of the duplex stem is very important because it will affect the positioning of the tertiary contact if in larger RNA domains where the magnesium ions are always abundant. So here we notice that the profiles, the WAXS profiles are very different for different RNA conformations. The peak or dip position shifts or they smear. All these variations must have something to do with the three-dimensional structure of the RNA duplex. So we kind of handcrafted these features in the WAXS profile as a minimum and maximum position or contrast. We use min or max because it indicates the destructive or constructive interferences of scattered X-ray photons. We then did some structural analysis of the RNA duplexes, extracting some important information as helical radius, twist, major groove geometry, etc. And we found these two correlations between the features in the WAXS profile and the structural information as labels. So the maximum in panel A, the maximum position is inversely correlated with helical radius, which is kind of intuitive because as the helical radius increases, the feature shifts to lower Q, corresponding to a larger length scale or lower resolution. But here, the helical rise might not be that intuitive. So given the complexity of RNA structure and the physics of X-ray scattering, we need more powerful tool to understand and model the WAXS profiles. And now imagine that there is an intelligent WAXS creature who can recognize these profiles and tell you the information you want. For example, this creature might look at the green profile and tell you that the helical radius is about 10 angstrom. And it will also be great if this creature can tell you about how it recognizes those profiles. And this creature is called machine learning. We use the RNA structure and WAXS profile to train the machine learning model and use the trend model to get information from the experimental data. And we call this deciphering. So the deciphering pipeline is the following. We use unbiased MD simulations as confirmation sampler and compute the profile and extract structural parameters for every one of the confirmation. And the WAXS profile are the feature vectors and the structural parameters are the labels, such as helical rise or group width geometries. Some of these labels or some of these parameters are continuous and some of them are discrete. For each structural parameter, we can train a machine learning model using the data splitting on the right. And once the model is trained, we have one synthetic data set and two experimental data sets sitting in this unknown box for the trained machine learning model to decipher. And if the model is trained well, the model should output a prediction of interest, even though there might not be a single confirmation that agrees with the experimental WAXS profile. And we will mostly focus on the model interpretability to see how, what kind of information the model actually learns. And based on the previous seen linear correlation, I will talk about a generalized linear models and move on to nonlinear models. So the linear model actually takes this form, y equals x beta plus noise, where y is the structural parameter and x is the WAXS profile. And beta is the coefficient and will reveal which features or which angle is important in the fitting. And there are three generalized linear models, and the difference is mainly in the regularization term. And I want to focus that the lasso takes the L1 norm as in the regularization term and focus on the sparsity of beta. And the training, validation, and testing errors are shown here. They look kind of okay with 0.01 mean square error, but not as precise as the nonlinear model I will talk about. Where the MSE, the mean square error, is about 1/1000th of the linear model. So therefore, the linear model might be just too simple considering the complex mapping of the three-dimensional structures into one-dimensional profile. And we need a nonlinear model with model interpretability. And a nonlinear model is the extreme gradient boosting model, or the XGBoost model. And the XGBoost model is basically a decision tree based model and can be used for classification and regression. So let me went off track a little bit to consider a problem predicting who in the family members prefers or likes the computer game. And you might come up with a decision tree of age less than 20 and assign different scores to each group. And then you might also come up with another decision tree where a person uses computer daily or not and assign different scores. And now you have an ensemble of decision trees, of two decision trees. And now for this boy, he gained 2 and plus 0.9 from the first and second decision trees, while for his grandpa, the score is minus 1.9. So we can, based on these scores, conclude that the boy is more likely to like computer games. So this process might go on, and you have a forest of decision trees. And note that there are two kinds of importance here. One is the weight importance, which means that the number of times a feature is used in a node to make decision, or the decision-making process. In this simple case, where age and daily use are show up in tree 1 or in tree 2, they are of the same importance, the same weight importance. And the other is a gain importance, or the score in the leaves, showing how determining or how important a certain feature is. And the XGBoost model is very versatile in physics, astronomy, finance, and many more, and we will apply this model to the WAXS profile. We use the WAXS profile as a feature vector X and structural parameter as label Y again to train the XGBoost model. And we also consider the noises in the training data and different number of features in the profile, because a WAXS profile is essentially a one-dimension curve, and a one-dimension curve can be represented by an arbitrary Q points along the Q axis. And this is also known as feature reduction or Shannon sampling limit, and which I will talk about. The performance is benchmarked against the random label here, where there's no pattern or no structural signals for the model to learn at all. And I only list helical radius and helical twist in this table, and all the models have great performance judging by the low mean square errors. And we apply the trend XGBoost model to the real experimental data with experimental noises, and the output is a distribution. And this distribution is actually the propagated error from the trend XGBoost model. And notice that the distribution are centered at the known or true values, demonstrating the success of these XGBoost models. And therefore, this method can be used for different systems, different molecules, and also different training strategy or different models might further improve the accuracy. But as a physicist, I'm not going to stop here because I'm more curious about what the model actually learns. And whatever knowledge it learns, we also want to know. So we can first look into how the model makes decisions by looking at the weight importance. And this is looking at the decision tree node as shown here. So for all the structural parameters studied and the different model strength, the importance traces are essentially very similar, and also notice that it's angle dependent. And we only train this model using the fine structural parameters such as helical radius or helical rise. And those parameters are beyond the resolution of small angle. So you see that the small angle traces are kind of irrelevant in the weight importance. And however, the main difference is in the gain importance. So although these models go through the same decision trees, the model assigns different gains or scores to learn different structural parameters. Some Q regions are extremely important. For example, in this case, the first wiggle seems to be very important to determining the helical radius, while this first local minimum is very important in determining the helical rise, and so on and so on. So therefore, we see that the structural information or the structural signal is very strong in the WAXS profile, despite of its physically and experimentally weak signals. So this interpretable XGBoost model can also build our intuition for qualitative interpretation by looking just at the WAXS profile. So after seeing the success of the machine learning model, there are three critical questions we are especially interested in. So how many structures do we need to train a good model? And this, the answer depends on the model accuracy. And, but generally about 15,000 is a good number. And how does noise in the training data affect the model? And here we see that for less than 10% of the noises in the training data, the trained model will still be acceptable. And lastly, which is my my personal favorite, how many Q points are needed for training or to representing for representing the WAXS profile? And this is also known as the Shannon sampling limit of solution accurate scattering. And in our case, this Shannon limit is the red line. And as you can see that the mean square error starts to surge if the WAXS profile is physically undersampled, losing a lot of important correlations or structural information in the profile so that the XGBoost model couldn't learn or performs poorly. So after talking about a genetic algorithm-based and machine learning-based method, I would like to focus on the molecular dynamics simulation a little bit. And here we use WAXS profile to improve the MD simulations on the RNA using a strategy called sample and select. As I mentioned earlier, WAXS profile is unique for different conformation, and it is the fingerprint of the RNA conformation with resolution higher than 4 angstrom. And this piece of data is very crucial for the molecular dynamic simulations because it is, first, it is in the solution, and second, it provides more than just a number. It provides a highly convoluted one-dimensional profile. And thirdly, it can probe deeper or probe larger molecules than the solution MMR. And there's no need for fluorescent labels or isotope labels. And we use MD simulations for training and sampling previously, but actually MD contains valuable physical models known as the force fields. But however, MD has a notorious sampling issue, which means that starting from a structure, MD simulation might take very long time to converge. And even though it converges, it might converge to the wrong conformation because the system might be trapped in a local energy minimum. And there has been many studies showing that the nucleic acid force fields have special artifacts. So we propose a better way to do MD simulations incorporating the WAXS experimental data. And this is an ongoing and exciting collaboration with Serdal and Weiwei and NYU Abu Dhabi. And we call this strategy sample and select. So we start from a conformation, an RNA conformation, and run the simulation at room temperature. And we check if the conformation matches the experimental WAXS data, and if so, we hit the jackpot because everything is working well, there's nothing to do, and which is seldom the case. But if not, we have this feedback loop. We run an MD simulation in higher temperature and cluster those structures, and this is a sampling step. We then check the cluster structures and select the closest conformation to the experimental WAXS profile and use it as a starting conformation again and again. So this is the selecting step. So in practice, a conformation can go through this feedback loop many times until convergence. And this method will allow us to efficiently and effectively search on a rugged RNA energy landscape, while the classical MD simulation or the brute force MD simulation might take a long time to find the wrong answer. And we apply the sample and select on the RNA duplex, on the DNA duplex of different sequences, the pure AT sequence and mixed sequence. And experimentally, these WAXS profiles are very different, suggesting different DNA conformations in the solution. And the MD simulation actually did not require sample and select. So we hit the jackpot in this case. The dashed line in the is a canonical B form, and it kind of agrees with the mixed sequence DNA data, while the AT DNA has a very different conformation than the B form. So this is the final MD structure, and the AT DNA, obviously, it has like a larger major group width and tighter minor group width. If we do the structural analysis, this is what we're seeing. It deviates from the major group average value and also the minor group average value. And then we further apply this on the RNA duplex in different salt. We try 400 millimolar potassium, 100 millimolar sodium chloride, and 10 millimolar magnesium. And you see the experimental data are very different, especially for the first peak right here. So we know that we can say that the RNA duplex has different conformations in the solution at different salt. So the gray profile are the results with of MD simulation without sample and select, and starting with the canonical A form duplex. And this canonical A form duplex is depicted as a dashed gray curve, and the A form does not agree with the experiment. But however, with the feedback loop of the sample and select strategy, we enhance the sampling at higher temperature, and we funnel down and select the conformation, and it converges to the closer conformation as shown in the black curve. And the final RNA structure are shown here at different salt concentrations. And these, as the concentration of monovalent increases, the RNA duplex start to show this kind of end bending or end effects caused by an ion-preferred GGG tracks at the end. And these simulations in the RNA and DNA, those simulations were run with a similar timescale, and the sample and select is needed because RNA energy landscape is rugged. So the starting conformation might direct the simulation into some local minimum. So the marriage between WAXS and MD simulation has many advantages. So our sample and select strategy enhances the MD simulation or MD sampling by 10 to the third to 10 to the fifth. Classical timescale or the brute force timescale is about millisecond, but now we can run nanosecond simulation to find the answer. WAXS is a high near atomic resolution technique in the solution and works great for RNA. And in the sample and select, the force field remained intact, which means that the simulation is essentially unbiased. And this team up between WAXS and MD simulation has many future potentials. The WAXS profile can be used to benchmark, to optimize, or validate empty force fields of nucleic acids. And WAXS data can further drive the simulation and overcome the sample and select issues and find the correct conformation of RNA. And this is a work in progress. So now let's take a step back and think about one question. All this success depends on atomic structures from molecular dynamics simulations. And we cannot just measure something we know already. Some systems are very hard to model and are even unknown to us. MD simulations usually do not work for those cases. So what can we actually do to get insight from just the experimental profile? So the problem is, can we actually get three-dimensional electron density distribution from the WAXS experimental profile without any prior knowledge of the molecular system being measured? And this is a famous inverse problem from 1 dimension to 3 dimension, also known as sparse data reconstruction. And if we do this blindly, we might create new information and introduce human bias. We need to condition the solution even better. We have to concede that seeing the atoms or seeing the RNA bases might be impossible because the phases and orientation information is lost in the solution X-ray scattering. So however, I will propose a method called frequency matching, refining the electron densities in 3 dimensions and also in vitro. And this method is inspired by dense, which I will talk about, and cryo-EM linearization, and also black hole imaging. So I would like to talk about dense first. So with just small angle experimental data or SACs, we can obtain 3-dimensional dummy atom reconstructions which represent the uniform electron density in 3D. And Professor Tom Grant at UBuffalo proposed a phase retrieval and shrink wrap algorithm that can reconstruct the low resolution, non-uniform 3-dimensional density. And notice that this is the result when dense was applied to different proteins, and you can see that this is still a low resolution technique because it uses SACs, but you can see some regions are electron dense and some regions are kind of electron sparse. And this is a very critical step for preconditioning our inverse problem because it narrows down the solution space from 3-dimensional real space to only within this 3-dimensional envelope. So with dense, we obtain a 3-dimensional shape. Let's just call it a potato, representing the excessive electron density in 3D. And I would also like to remind you that the scattering angle Q corresponds to the real space resolution. The higher Q you go, the higher real space resolution you get. And the idea of frequency matching is actually carving this low resolution potato using an array of spoons of different sizes. So the idea of frequency matching is the following. We have a WAXS profile. We go to a Q equals Q1 corresponding to a low resolution and a large spoon. We use this spoon to carve the potato, then we march to higher Q and Q2. And Q2 corresponds to a smaller spoon, and we use this smaller spoon to carve the potato again. And revealing finer and finer structures as the spoon gets smaller and smaller, and higher Q is marched. And finally, we will end up with a finely carved potato, represent the 3-dimensional electron density and structures, and then we are done. So in practice, this is how the frequency matching looks like. You start with a smooth 3-dimensional shape, and this is low resolution because we only use small angle data. And the frequency matching algorithm will match into higher and higher angles, and the structure becomes finer and finer. And notice that those fine structures was originally invisible in the small angle X-ray scattering, and only if you go to wide angle X-ray scattering, they start to show up. And the key point here is that the result from each frequency matching step conditions the solution better for the next frequency matching step. And here is the pseudocode for the frequency matching algorithm, and we need to scheduling a lot of the parameters because as we march to higher and higher Q, we need higher and higher precision. And the core of frequency matching is actually in this for loop. So we think, in this for loop, actually the algorithm marches higher to higher and higher Q. And within each frequency matching step, it smears the density a little bit while maintaining the overall shape from the last step. And the frequency matching then splits the density into finer grid or finer voxels corresponding to the resolution. And it starts the carving process, which is essentially an optimization process. So here's how the carving process looks like. And for the smooth shapes, some structures within were carved out. And as higher and higher Q is achieved, these structures start to show up. And I want to emphasize it again that this kind of fine structures were originally invisible in SACS. So in SACS, what you get is a low resolution reconstruction of the envelope. As you march to higher Q, these fine features will appear. And notice that this blue shell around the molecule represents the solvent and ion shells with higher electron density compared to the bulk solvent. And we applied the frequency matching on the real DNA WAXS profile. As the algorithm marches to higher and higher Q, finer structures, for example, like the holes, electron sparse regions, or the phosphate backbone arrangement, they start to appear. And there are two plots here. One is the electron density map with rainbow coloring, and the other is the wire plot. And this wire plot just takes the electron dense voxels out, so you won't be confused by those coloring in the density map. And notice that only if you pass 0.33, which is a small angle limit, you start to see those finer structural details from the phosphate backbone or the bases. As you march higher Q or higher resolution in real space, those features or those phosphate backbone arrangements is still the same, but you have potentially smaller and smaller voxels. And we further apply this to a molecule called MELA1, which is a cancer-related RNA triplex. And the triplex region is only near the bottom 2/3 of the molecule. And this is why this this carved structures seems more electron dense near the bottom. So you see that this part below is like more electron dense, and this is due to the RNA triplex. And the agreement with the experimental data is good up to about Q equals 1.5, which is the record as far as I know. And the docking between the density and the real structure also confirms the feasibility and the power of frequency matching, also the WAXS experiment. And this is the application on a structured RNA. How about flexible RNA systems? So for flexible RNA, we use single-stranded RNAs. And for the single-stranded RNAs, we don't see any obvious WAXS features in a high Q region. And the carved result looks like a randomly distributed electron density in 3 dimensions. And if you further extract those electron dense voxels out, it doesn't look like anything because those single-stranded RNAs are intrinsically flexible in a solution. And therefore, the frequency matching algorithm is best for structured and stable RNA conformations with possible application to other macromolecular systems such as lipids. And finally, I would like to conclude my talk by bringing back some of the problem statement. So given experimental and theoretical RNA WAXS profile, we can actually learn very detailed structural parameters such as helical radius, helical twist, major groove geometries. And those parameters are originally invisible in a small angle. And we can also bridge RNA conformation predictions and WAXS measurement using extended ensemble optimization method, which is a genetic algorithm or machine learning. And we can also improve MD simulations or tackle the MD sampling issue by using the experimental WAXS profile via a strategy called sample and select MD simulation, or even WAXS-driven MD simulations. And without any RNA or without any atomic structures at hand, we can apply frequency matching algorithm to the experimental data only, and we can see the structural periodicity or phosphate arrangement by the molecular system. So here's the two-dimension map that shows my PhD journey. On the left, we have without or with atomic structures, and on the top, we have small and wide-angle X-ray scattering. So we use the SACs initially without atomic structure to understand RNA junctions, and we include atomic structure in the SACs to model RNA junctions, and also now we are working on a project cross-validating with X-ray crystallography and also cryo-EM. We further push the boundary into an uncharted land, which is the wide-angle X-ray scattering. We need atomic structures for modeling and deciphering the WAXS profile using genetic algorithm or machine learning. And we further use WAXS to better empty simulations by sample and select strategy. And finally, we march even deeper in the frequency space and utilizes only the WAXS profile, only the experimental WAXS profile without any background knowledge of the RNA system being measured using the frequency marching algorithm. And here I want to talk about some future potentials and future aspects. So on the theoretical front, more accurate theory on the excessive density scattering theory is needed to push the Q beyond 1.5. And also, we need to match the theoretical sample concentration with experimental sample concentration by a proper choice of empty simulation box. And this field, this is a very new field, and it will depend heavily on machine learning for reconstruction, recognition, interpretation, prediction, and many more. And also down to the very fundamental physics of solution X-ray scattering, people have shown that global structures such as radius of gyration or molecular dimension can be expressed as a linear combination of the harmonical coefficients, or the coefficient in front of the Fourier series. But as we go deeper into the wider angle and finer structural parameters are revealed, no one has shown that if this is still true or not. On the experimental front, time resolved experiment or temperature jump, pH jump of interesting RNA system will reveal the kinetics of RNA and build our understanding into interesting RNA systems. And experimental WAXS profile can be used as a benchmark validation or correction for empty force fields of nucleic acids. And also, these WAXS profile can also serve as a cross-validation data for other biophysical measurements such as cryo-EM or X-ray crystallography. And finally, this field is geared towards structural-based drug design and also structural-based drug discovery. And acknowledgement, I want to emphasize that this work is mainly grounded on the advanced beamline development. So my special thanks to Richard, Jesse, Arthur at Chess, and also Shirish and Lin at NSLS2. And also thanks to the NIH funding and Cornell funding and all my collaborators now and then. I also want to thank these facilities, Cornell Chess and NSLS2, and also Cornell Red Cloud. I also benefited a lot from the support of Julia and C++ communities, and I'm grateful for my community members, Sumanta, for his input in machine learning, and Warren for his input in my single molecule thread optical setup and also RNA sample labeling. And finally, I'm forever thankful to Lois because of her patience, intelligence, kindness, and research freedom. And I was more than lucky to join her group and meet these brilliant, brilliant people. And with that, thank you, and I will be happy to take questions.
Sorry, I was muted, and I'm applauding, so that's good. Thank you, Yen. Okay, that's thank you very much. So, are there any questions from the public audience? Jan, please.
Sure, I can ask on. Excellent talk, really just, yeah, fantastic, very impressive work. So of course I do have to ask a question. So I was very intrigued by the machine learning, and I guess one part that's always a big discussion is about the training set in terms of how big the training set has to be to get robust results. For example, facial recognition, people found really good at recognizing white faces, but then diverse faces, minorities, not so well. So what do you need kind of to have a good training set for your RNA? Do you need to just change the concentration, the sequences, the length of them?
Yeah, so we focus on one single construct. So this one single construct is intrinsically flexible in the solution. So it has potentially many different conformations in the solution. So the training data, we don't include the sequence variants, but we take into account the salt-dependent RNAs. So those are, and also we only care about the three-dimension orientation or arrangement of the phosphates. So we think about 15,000 RNA conformation should be good enough for training a good machine learning to decipher the experimental data and learn something from it.
Great, thank you.
Yeah, thanks.
Okay, anything else from public participants? All right, so why don't you guys all give Yen a hand? It can be